Good morning, everyone. Welcome to the second day of the Natural Products Applications Congress. This morning will be dedicated to the cosmetic area, and before the break and poster sessions, we will have the opportunity to hear 2 keynotes and 6 oral presentations. Before that, I would like to remind you that you can see the posters of each area, food, health, and cosmetic, in the corresponding link at the website of the conference. And also during the presentation, you are invited to ask some questions on YouTube live stream chat so they can be answered at the end of each presentation. And now it is my honor to present Dr. Ana Palmeira de Oliveira from the University of Beira Interior. Dr. Ana is graduated and master in pharmaceutical sciences by the University of Porto and PhD in pharmaceutical sciences by the University of Beira Interior. She is invited professor at the University of Beira Interior, researcher at Health Science Research Center, and CEO at Health Products Research and Development, LabFit and Pharmapoly. Dr. Ana completed her master in business administration at the University of Beira Interior in 2018 and is author of more than 60 papers, 1 book chapter, and 3 patents. Her work has been recognized with 8 prizes. Her research field is in the health sciences, specially dedicated to the study of skin and mucosa disorders, mainly in the study of bioactive compounds as cosmetic ingredients and pharmaceutical actives against pathogens, the host pathogens interaction, and the safety assessment using in vitro tests. Dr. Ana, thank you for your availability to share your valuable work entitled Natural Cosmetics, a Market Trend Sustained by Science. Whenever you want, Dr. Ana.
Thank you very much. Thank you very much, Carla, and thank you also to the organization committee for this very nice invitation to be here today, sharing with you our experience, my experience as researcher, but also in our position in marketing the company, how we also relate with these issues on natural cosmetics and ingredients and consumers' expectations for the new products that they demand to be developed. So this is a combination of perspectives and experience, and I hope at the end I can contribute to elucidate more than confuse you. So in fact, when we look to the market, we see that consumers' demands in the last decade, more than 1 decade now, but they demand for products that are considered natural products. And this is more than the natural composition of the product. So consumers, they like that products have a whole concept of being natural. And this whole concept is related, of course, with the composition, chemical composition, but also with this concept of being products that are eco-friendly, that are aware of the animal welfare policies, that do not use packaging too much, or this packaging is also biodegradable, and also this very important issue that is the capacity, the ability of these products to give them the experience of being revisiting traditional use. So in fact, when we look to the consumer demand on natural products, we have to consider that natural concept is behind natural composition. So when we develop a product, when industry develops a product, and also when researchers do contribute to this natural concept of cosmetics products, need to be aligned with these expectations. Of course, that natural composition is very important, and it leads the industry to the need to bring raw materials and products that are based on natural composition and that have biological certification. So this is really a main issue related to the ingredients and to the capacity that we have to bring new ingredients to the market. And these ingredients being very well characterized. This is, in fact, based on a lot of sciences, and all the other demands of the consumers related to natural concept also. It is a lot of science behind this natural concept. And this is something that is the focus of my talk here today, and the sharing, the process of sharing with you the experience that we have. So this is so important that there is an ISO published defining what is a natural product, what is organic cosmetics, in order to regulate what at least these cosmetics have to have in their composition and the origin of these ingredients. But when we look to the market, and recently me and Rita, she's my sister, we supervised a master student in pharmaceutical sciences, and we looked to the Portuguese market, and we checked for 300 products that have in their labels something related to cosmetics, natural cosmetics. Diana was the student, and she made a wonderful job doing this analysis of the market. And at the end, we concluded together that in fact this is confused. A lot of certifications are available. I will not spend too much time in this, because at the end of the morning you will have a talk on this issue also. But well, I would like to share with you that our thoughts was that one symbol for international certification will be really a very interesting approach in order to clarify and make it clear for everyone this communication on natural compounds. You know, the depression that the industry has about the natural cosmetics is really based on the ingredient and the quantity of ingredients and the diversity of these ingredients, and how safe and how active they have to be competitive in the market. So this is something that industry and researchers have been investing a lot during the last years. But at the end, we need to keep in mind that despite consumers' demands for natural products, one idea must be consistent also in the market, that natural does not mean safe or better. So the ingredients, the new ingredients that we propose, both in the researcher perspective or already in the industrial perspective, must to be very well characterized, both on their bioactive approach and also on their safety assessment. And this is something that in the market is very frequent. People thinking that for being natural, things are better or safer. Usually they are more active, and if used with some control, they are of course safe. But it is our responsibility as researchers to find those that are active and those that are also safe. And this was something that was a central point in the cosmetics regulation that changed in 2009, and that was started to be active and started to be mandatory in 2013. And this changing in the regulation was a lot to do with the safety issue too. So the regulation put it very clear that all the products that are in the market must be safe. And for that, there is an international scientific committee that gives opinions and that defends the safety of these ingredients, and also that demands that the ingredients that are not safe must be removed from the market. So this is the scientific committee that is behind the safety assessment of cosmetics ingredients. So there is a lot of science in this pathway. All the ingredients, as I said, must be this pathway to the market to prove their efficiency and to prove their safety. And don't forget that consumers have a high expectation. They really want that natural ingredients show to be more efficient and more safe. But for researchers like us, natural ingredients can also be challenging at labs because they are, most of them are mixtures, and to test mixtures in the safety approach can be, in some cases, in some tests, can be very challenging experience. When we plan research projects, or when we plan to prove or to test if a new extract, a new ingredient, natural ingredient or non-natural. Okay, this applies to all the ingredients, but focus on our approach of discussing the natural ingredients. When we look to the plan of planning a research for a new ingredient, we need to keep in mind that the regulation also brought this change, that is the complete animal testing ban. So the tests for safety or efficacy cannot be made in animals, just on in vitro or in vivo non-animals, so in humans. And this also stressed a lot of research. And this research for new methods that substitute the in vivo animal methods, so replace the animal methods for non-animal in vitro methods, is validated by a group of European labs that are responsible to prove, to test, and to qualify a method that is proposed. A lot of methods are proposed for researchers that are in the universities, that are in the research centers, that are in the polytechnic institutes, and that want to propose a new method to replace an animal method. So this network of labs validate these methods, and if they are valid enough, they are robust enough, they will be described in a procedure, in a SOAP procedure to be used worldwide. So this is also a research and investigation approach. For instance, one example is the eye irritation methods. You all know the Drace test on rabbits that was replaced by in vitro methods. Now we use the HAG method for chicken. It's a non-animal method, or the bovine corneal opacity, or the skin model, sorry, it's not skin, but the three models, tissue models for eye replacement. It's also possible to use 3D model for eye replacement tests. So this was the result of a lot of research on the cosmetics field that is very important, and that is a continuous process, because we continue needing new methods, and it's really, really important, because to test new ingredients, we need to have robust methods. We need more tests, but we already have some answers for some of these topics. For instance, for skin corrosion, skin irritation, there are already 3D models that can be used to characterize the safety of new ingredients, and that can be used for natural ingredients. We are very used to use these methods, so we have experience with these methods, and they are easy to be used, and they are very robust, so they can be useful for you if you want to change, to check and to test for new ingredients. There are some cases in repeated dose toxicity. For instance, we continue having some problems with the replacement, but well, the pathway is going, the research is ongoing, and I believe that soon we will have a complete replacement. I invite you to check for these two posters that our team is presenting in this Congress, and that where you can see some of this, the use of these methods to characterize plant extracts. But the concept for the consumer about the natural product, as I shared with you in the beginning, it's not only on the composition or in the safety of this composition for the human use, for the human skin or for the human mucosa. Is also about the environment perspective. So biodegradability and ecotoxicology are also very important issues when you test for a new ingredient. And the natural ingredients have this challenging of being even more biodegradable and even more eco-friendly, eco-safe, because this is what the consumers are demanding, and this is the consumer expectation. Celso Ferraz will also present some of our results on ecotoxicology evaluation of plant extracts, and you will be able to check for the use of a method here at our university. But of course, that the ingredients have to have some efficacy, or they have to have a new value in the market. They have to have added active value in the market. And one of the most frequently tested is the antimicrobial effect against protecting human from pathogens. So the antimicrobial effect against human pathogens is very frequent to be published with natural ingredients. And we tested for some, for instance, natural mineral waters from the central region of Portugal. We tested the antimicrobial activity, and we published this information. And also there is an important paper from larva extract. There is another approach of natural ingredients, well, the most frequently tested, that is very important, the antimicrobial properties of oils used in cosmetics, both vegetable essential oils and other extracts that can be used and proposed as ingredients. But also, one of the things that we see in the market in the last years is this approach of claiming preservative-free. And consumers have also some expectation that a natural product do not have preservative, a chemical, synthetic, as they usually say, chemical. Well, natural products are also chemicals, but synthetic, they want to say, preservative in the formula. But about this is a tricky thing, because the regulation that I shared with you in the beginning of this presentation is very clear. There is a list, a positive list of preservatives that can be used in cosmetics. And of course, we can contribute with new preservatives. We see that, for instance, plant extracts, essential oils, if they are present in a formula, as this review states very well, if the essential oils are present in the formula, in a cosmetics formula, they have the natural capacity to preserve the formula because they have the antimicrobial activity. But then we have to keep in mind that the regulation is clear, and the preservatives that are listed in the annex, the positive list, are the ones that have to be used, the only ones that have to be used. And for instance, in Portugal, our regulatory entity, the position of the Infarmed is that all the cosmetics products that do not have an exemption of using a preservative, meaning cosmetics product, for instance, that have water available in the formula for microbiota to grow, they have to have an addition of a preservative that is in this positive list. So they do not allow the replacement of the preservatives that are listed in the regulation from using other extracts or other ingredients that could be used, that could be having activity and effect of preserving the formula. So for regulatory entities, the preservation of the formula must be done using a preservative that is in the regulation, so that is in dispositive list. So we can contribute for new preservatives in this list, including the approach of testing natural compounds. But the dossier must be produced, the tests must be done, and the dossier must be proposed to the SCCS committee that I shared with you in the beginning. Other efficacy tests are also proposed on natural ingredients, the antioxidant effect, the anti-pollution effect, now related to the pollution that we have. For us, in Portugal, we don't really have a lot of this problem, but in other countries, the anti-pollution effect is really a very important claim. But also the blue light protection. We are all submitted to blue light effect using our laptops, using our mobile phones. So this is also a very important trend in the future. If we combine different tests, we can support a new ingredient for instance, acne. For acne, it is important the antimicrobial effect of the new ingredient and the antioxidant effect of this new ingredient. The antimicrobial effect, for instance, against Propionibacterium acnes, that now is called Cutibacterium acnes, and also against Staphylococcus aureus, Staphylococcus epidermidis, of course, but also the anti-inflammatory effect of these ingredients. And I also invite you to check for this poster where we share with you some of our results on anti-inflammatory effect of two Timo species from Portugal. Another important trend that is also based in science and that is now producing a lot of data and discussion around is the microbiome trend, the microbiome claim. And for natural ingredients, what consumers are expecting is that the natural ingredient protects the microbiome. That the natural ingredients do not have a significant impact in the microbiome in an healthy skin, of course, because if we have a disturbance of the skin, like in acne, we want the natural ingredient to have a role in controlling the microbial growth. But when we are talking about healthy skin or healthy scalp, we want, the consumers want the natural ingredients not to contribute to disrupt the microbiome. And what is this about the microbiome? The microbiome is the collective genome that we have in our skin related to microorganisms. So it's the sum of the microorganisms that are present in our skin in each moment and those that are transient, that are microorganisms that in some moment, for some reason, they are there and they are temporary. And as I don't know if you have this idea, but the skin microbiome, the microbiome, and now we are talking about skin or scalp, the skin microbiome is stable over time. With some external exposures, the microbiome can change, but if the external exposures stop, the microbiome will return to the natural, to the original profile. And it is established, it looks like that it's not in the birth moment, but in the placenta. And this is something very new too. This is something that was very recently described. So we need to protect it during our lifetime, and natural products can contribute to this. And also the perspective that not only the microbiome protection is important, but the relation of the person with the environment. So this perspective of being biodegradable and being safe for the environment. Again, the all or beyond perspective. So this is really a good opportunity for applied R&D in this field of new ingredients. These are some publications that we contributed with antimicrobial tests, for instance, new ingredients that were proposed. And also, I would like to share with you and invite you to visit the website of this R&D project that we are promoting together between our company LabFit and our university and the Polytechnic of Castelo Branco, that aims to test extracts, natural extracts from plants produced in Portugal, has in this perspective of testing the safety and efficacy of these extracts, promoting them as potential cosmetics ingredients. So this is a project that we will finish at the end of this year. We already have good results, and I invite you to come to visit the website to have more information. It will be updated soon. We tested and we are studying this species, and we are very enthusiastic. We are very happy with the results, as some of those are really very interesting ingredients for cosmetics industry. So this was our perspective that I would like to share with you, and I'm free now for answering your questions.
Thank you again, Dr. Anna, for your interesting presentation. We have at the moment no questions from the audience, but I have a curiosity or questions, I don't know. What do you think are the most urgent steps to overcome in the application of natural products in the cosmetic area? Do you think it is the research? There are science limitations, because you said that this is related to several fields of research, not only the ones that you talked about. Or do you think it is most the legislation steps that need to be overcome and the unifying of this legislation to be common to all the countries?
Well, regarding the unifying of legislation in Europe, we already have the regulation. So we are already in a good condition to promote these ingredients. Regarding science, we already have good models. Of course, that we need some models, but this is an ongoing process. It's something that we continue. All the scientific community continue investing in this. So it's something that I think that every year we are better. What I think that is mandatory now is to promote the communication between the resources that we have in natural ingredients in the different countries, meaning the production process, for instance, the production capacity, and the industry that aims and that wants to use these ingredients. Putting in between the scientists that need to validate the natural extracts or the natural ingredients in order to industry to be able to use them. So I think that this triangle of three fields that are very important: the production, the resources that are naturally available, what are those, how much we are able to produce or to collect without interfering with the environment. What is the science that we have supporting these ingredients, and what are the industry needs and demands on the quality and on the information needed for these ingredients. I think that this communication is really what is missing, but it's not missing totally. We have good examples of natural ingredients that started in the research labs and came to the market and that are available to be used by the industry to produce the natural cosmetics. So I think that we can improve, but we already have good examples.
Okay, thank you very much, Dr. Ana. Now we have a question from Sara Gonçalves. When you formulate toners using hydrosols, most hydrosols come ready preserved when you purchase them from suppliers. Even though not all suppliers declare this. Do you need to add that preservative even so, sorry?
In the when the produce, I'm sorry, I didn't understand.
When you formulate toners using hydrosols.
Okay, you have to, well, at the end, in the cosmetic product, you have to make an important test, that is the efficacy preservative test. The capacity that your formula has to preserve the product from the growing of microorganisms. So the preservative efficacy test is something that you have to do in the final product that you developed to validate that your product is protected. If the concentration of the preservatives in the ingredient that you bought are enough to protect the formula, well, you don't need to add. But I think that knowing the concentration of the preservatives in the ingredients, I think that I may be saying that it's not enough. You will have to add additional preservatives to preserve your formula, but you must decide based on the preservative efficacy test and analyzing the concentration, final concentration of the preservative that you have in the formula that came from the natural extract.
Mm-hmm. Thank you. And now Elena Ribeiro also has a question, if you don't mind. What does it take to incorporate an ingredient into the market? For example, a natural ingredient in cosmetics.
A new one, or a natural ingredient that already has authorization to be used in the market? Well, if it's a new one, you have to characterize the efficacy, the safety. You need to combine this information, and if it's really, really new, you have to have a safety opinion on the ingredients. Because it's important to validate the ingredients. But if it is some ingredient that already exists, that is already used in cosmetics for a particular production, for a specific production of this ingredient, you have to have your own characterization of the ingredient, your technical sheet of your product. Meaning that you have to describe what you have in this ingredient, including impurities, if it is the case, for instance, and you have to have a safety data sheet for that ingredient. I don't know if was this the context of the question.
Yes, she wanted to ask for a new one. Yes, thank you for your answer. And lastly, we have a question from Isabel Gaivão. Are there approved assays to test genetic effects on natural ingredients?
Yes, mutagenicity, for instance. There is a test to test the effect of a new ingredient in the mutagenicity genotoxic safety approach. The AIMS test is very widely used to test genotoxicity, but there are others, including 3D models for genetic toxicity.
Okay. Thank you very much, Dr. Anna, again for your availability. And I hope you're enjoying the Congress. Stay with us, please.
I will stay. Thank you very much. Thank you.
You're welcome. And now I'm honored to present Dr. Paola Perugini from the University of Pavia. She is a PhD in pharmaceutical technology and is a professor in the pharmaceutical and cosmetic technology at the Department of Drug Sciences at the University of Pavia since 2001. She is director of the second level master course in cosmetological sciences at the University of Pavia and is also founder and scientific director of Etica SRL, academic spin-off of the University of Pavia. Her research fields concerning pharmaceutical and cosmetic technology led to several patents and more than 150 international publications and Congress publications. in cosmetic field, Dr. Paola received the Maison de Navarre Award for research regarding studies on photostability of sunscreens. Dr. Paola, thank you very much for being here today, presenting your presentation, your collaboration entitled Upcycling, Transforming Byproducts from Waste and Agriculture into Active Ingredients for Cosmetic Use.
Thank you very much for your kind invitation and for kind presentation. Today I'm here, I'm very glad to be here and to speak about one of my main topics of research, because I'm in the university and together with the spin-off ETICAB, we work in cosmetic development, new cosmetic, but particularly concerning the efficacy and in vivo, especially in vivo efficacy test to produce, to obtain a very stable, safe and efficacy product. So today I'll speak about upcycle. Upcycle is a good word. It's a very, very meaning in the world, because upcycle can be defined as a process to permit the conversion of waste material to something useful or valuable. I think that upcycle sometimes can be confused with other terms, and for this I would ask and would thank also my colleague that speak about before me, because she made a very interesting presentation about.
I'm sorry, Dr. Paola, to interrupt you. Can you please put the presentation mode?
Oh, it's already that, but maybe I wrong the.
We are seeing the presentation. It's just to put it in the
sorry.
presentation.
So I do.
Thank you very much.
Yeah.
No, it's
I'm
not
sorry.
yet. Not yet. Sorry.
Okay, so, like that. It's better.
Yes. No,
Okay.
it's fine. Thank you.
Okay. Thank you. And so I would like to thank the Dr. Hannah Palmera, because she talked about natural product and sustainable product, but upcycle is different. So it's very important to talk about this term, because the upcycle can involve a different field, different several applications, cosmetic, but not only cosmetic, of course. Especially, I would like to point to you particularly in Italy, we have an area that we have a leather chain production that use all piece of leather coming from animals to from bitten to many different products. Also the hydrolyzed collagen to produce fertilizer for plants. So upcycle is the use of all type of waste coming from different type of production. I think that in cosmetic, so maybe, okay. In cosmetic, the trend zero waste born at the end, I think, of 2018, like a trend, because after the research to reuse the packaging for the huge devastation of our nature world, it was necessary to try a way to eliminate or reuse or readapt, starting from the packaging, but it's not only a packaging field. In fact, from a trend, zero waste, now we have a movement, a new movement in the cosmetic field, in which a new archetype for a beauty and personal care industry is developed. So it's very interesting, because consumer require more responsibility from brand. They want that brand be more environmental, more friendly with the environment, and take seriously the direction to the world and to the nature. So a lot of new research and new product can was be used in cosmetic film starting from the waste. There are some ingredients and some product that already used in cosmetic, like some oil, but from the product, the change was to produce new ingredients starting from the waste from food. So the food waste become a new ingredient for cosmetics. This is some example of recently a product coming into the market, like ingredients usable, suitable for cosmetic application. For example, an active hair care produced by Laboratory Expansises in France that developed some ingredient from damaged avocados. So we can reuse some very important fruit. Givaudan, with the startup Coffee Bueno, recently launched Copef as an alternative to argan oil in skincare products. So it's very interesting because we have a lot of waste from coffee. And the coffee is already very interesting for its activity. And the very new ingredients and very strange ingredients, because it's the first time for this kind of ingredient in cosmetic, is at the end of 2011, Dr. Croft launched a range of natural hair color based on waste blackcurrant pulp. This is very strange. This innovation received also prize because it's very, very interesting in new application. But we have to underline, we have to highlight that it is not so easy to use the waste of some product coming from food because we have a lot of process involved to obtain an ingredient can be used in cosmetic. First of all, different several methods can be applied to the matrix, to the waste, to obtain a mixture of ingredients. The process are the old or the new one processes, especially the supercritical carbon dioxide extraction that is very interesting because it doesn't involve the use of solvent, but some microwave go into the green chemistry, also for other application, and so on, a little very huge possibility for the process to obtain ingredients. But keep in mind that when you obtain an extract from a waste, you don't obtain only one substance, but you have generally a mixture of substances. And so it's very important to proceed with a path, to proceed with a very, very important path to obtain the substance or the mixture that you need to apply in cosmetics. So usually, what kind of ingredient can obtain from waste? Usually suitable ingredients can be divided in liquid, and very often you obtain lipidic phase like oils, natural oil, so can be chemically oil like a triglyceride, but not only ester with glycerol, but you can obtain ester, different ester, and we can name all oils also if there is some chemical differences between their structure. But you can obtain a liquid phase, oil phase, and a powder. The very interesting point in the recent years is obtain functional powder, because this is the way the strategy can be winner. The winner strategy is to obtain the best results when you starting from waste. And I want you to show, and why show you some example to understand what I mean with winner strategies. First of all, like the colleague of mine started talk before, there are a lot of steps you have to go across for obtain ingredient with high quality, high efficacy, and of course high safety for consumers. And a lot of steps are needed also, and especially starting from waste. You have, first of all, identify the ingredients and proceed with its characterization. And it's not always so simple, so easy. And then you have to evaluate the safety of the ingredients, and if it's not a new ingredients because you came from a waste, you know exactly which substances are inside, and all substances are well known from literature and from the psychological point of view. You can perform maybe only preliminary safety study in vitro, and then go to the safety assessment in vivo with at least with the patch test. Then you can evaluate the efficacy of the ingredients you obtain from a waste, and then you can formulate the product, the cosmetic product, and then test the final product. So it's a very long path, but it can give satisfaction if you can obtain. But if you want to start at the beginning, I want to show you that it's obviously also for ingredient coming from waste, you have to assure the requirements. The requirements required for all cosmetic ingredients: the safety, the quality, and efficacy. First of all, the safety. So you have to know exactly which substances are in the waste, and which substances you extracted, you have in your mixture, your product. So you have to identify. The problem from the waste can be related to the natural sources. So you have to, of course, like for all natural ingredients, test the absence of pesticide, and of course, the presence at the level of heavy metal, because maybe can remain in the product obtained from extraction, from the waste, maybe from the process, the use for the extraction. And also for this kind of ingredients, you have to produce the safety data sheet, in which you can put also the toxicological information, the eco-toxicological information also, and then the certificate of quality. The certificate of quality define the characteristics for each batch of product obtained. So it's not the same, it cannot be the same for all batches of ingredients, because sometimes we can have little difference presence of some substances inside, some color, maybe also some differences in pH, maybe in particle size if there is a powder. So the certificate is specific for each batch of production. This is for all ingredients. It's very, very huge important for ingredient coming from waste, because you can start it from very heterogeneous materials. So you have to standardize the procedure to obtain always the same ingredients. About the identity control of raw materials, we have to divide the technique that you can use to define which substances are in the complex, in the matrix. So you can use a chromatographic technique like CC or SPLC, or as you want, you have in laboratory to understand the type, to divide the single molecules. But it's very important for each batch of product to identify the mixture, the whole mixture, and the sampler method to be used to the identification of raw materials, and now approved also in the pharmacopoeia, the official test in pharmaceutical regulation, can be the near-infrared spectroscopy. The near-infrared spectroscopy can give information, very precise information about the ingredients, and especially for the reproducibility of the batch production, because the company gave and defined the threshold for the identity. If the new batch is inside the threshold, the batch is approved. In other case, the batch is banned. So near-infrared spectroscopy can be a chance for company to obtain very, very fastly the response about the control, so the identity, but also for the control of water content into the product, into the ingredients, because very often when you start from a waste, you have a high content of water, of humidity, and it is essential to dry the starting materials before obtaining the ingredients. And the check of the moisture, of the water content is essential. And the infrared is very fast method to obtain this result. You can also obtain concentration, a calibration curve, and you can also test the content like a percentage of water inside the dried product. Then I want you to show some example of the path about the use of some waste coming from nuts, especially oil coming from the shells of different type of nut, and their application in cosmetic. Starting from the pecan nut is a very nice nut, appreciated worldwide for their different for the taste, but also for a characteristic very important for alimentation, for food. And following the path we saw before, we have the first step of characterization of ingredients, especially for acid content. And we have a lot of unsaturated acid in all nuts, and this is very important for also for skin. About the safety evaluation, was performed the in vivo tolerability evaluation through the patch test, and these ingredients was defined as a no irritant, and no adverse effect was evident after application. About the efficacy evaluation, we try to evaluate two different types of efficacy. First of all, the efficacy against the radicalic activity. So we use the antioxidant activity test through the DPPH test. It's very common, very simple method to evaluate the antiradicalic activity, especially for lipophilic substance. And we use as a control the argan oil, very used in cosmetic field. And we made a comparison to the standard, to the control. The pecan nut oil coming from waste, so from the shell, is has demonstrated more activity against the PPH, so it's more anti-radical power to compare to the argan oil. In the same way, we studied pistachio. So the same way we obtain oil from waste, from the production of the pistachio cream for food, or pistachio fruit to be consumed. And also in this case, we obtain a characterization of the ingredients, definition of no irritant through in vivo tolerability test, and the activity was through the DPPH test was higher than the control 2, again, like pecan nut. Obviously, no pesticide and no heavy metal are present in all products we tested for in vivo. The third nut we evaluated was the hazelnut, so the oil coming from waste from the production of hazelnut and the use of hazelnut, especially in Italy for Nutella, is a cream based on the hazelnut fruit. In this way, we made the same path about the characterization in acid, the evaluation of no irritant effect on human. At least 20-25 people was enrolled for each test, and the evaluation through the DPPH. But after that, we wanted to verify the opportunity to use these oils in cosmetic formulation, and we produce the oil for hair care spray, for hair care, especially spray. The spray are common formulation for hair, and the presence of high content of oil inside the spray can claim several effects, like shining effect on hair, compatibility, so it's more easy to perform several form of the hair. And we want to evaluate also the antioxidant capability to protect hair after application. The antioxidant capability that before we tested only in vitro with DPPH, starting from the oil directly in cuvette and in spectrophotometer. Now we wanted to apply the test on hair. So we use the stress, and we analyze stresses, and we verify the activity, antioxidant activity, normalize for quantity of spray used in stress and for gram of stresses used. Then we verify the shining effect in vivo with a new technology. The new technology is a filament surface tester. Is a technology we developed in the university, and now we are patenting. We are under patent, European patent, attending the number of the patent. And it's very important technology because it is possible with only one instrument have directly the efficacy about the shine, the color of hair, and all in general, or filament, like all eyelash, tissue, or as you want. But about hair, this technology can give information about color, about shine, about combability, and curly and frizzy hair, but indirectly also about integrity of the hair. This is very important because if I want to use an ingredient and a product that can protect hair against against the treatment, hot treatment, against chemical treatment, maybe some permanent or some glazing product, or also protect against UV rays. We can give directly what only one instrument all these activity. So the results apply the same concentration of the ingredients in several sprays for hair in vivo, and analyze the hair compared to a control with the same ingredients like excipient, but we use argan oil at the same concentration as standard. So the results confirm that the oil coming from different nuts can have different efficacy. In particular, pecan nut oil gave a very high shining effect, more than 88% to respect to spray based on argan oil. The pistachio give to the spray a higher antioxidant activity, very interesting activity, and the hazelnut oil can improve the combability of the hair. So we have 3 ingredients with 3 different efficacy. By the way, I want to show briefly one other big field of application for waste, especially in Italy, and is the use of the residues, the waste coming from the wine production chain. It is well known that all part of the wine production chain can give important substance for different field of application. All of the ingredients of all substance are very known for antioxidant, microbial agent. The first ingredient studied in this field was resveratrol. Here you can see the structure. But our work is specific to try to use the pomace, because here in Italy, we have also in Portugal, but we have a lot of area in which we produce a lot of wine, and we have a lot of waste. So the valorization of this waste is a very important field for to find a new value for economic company. So in literature are a lot of reports concerning the efficacy and the function of different parts of the grape extract or the Vitis vinifera plant. It's very important for different parts can have different function can be suitable for cosmetic application. In our research, and then I can go to the end of my presentation so I can stay in time. We have a project co-financed by the Regione Lombardia with the aim to valorization of grape pomace. So we started to use the pomace after the first fermentation of the wine, of the grape. And after a drying process, we divide in two different process to obtain different substances. So we try to use the supercritical carbon dioxide extraction to have an oil fraction. This way it can be limited because it's a very low yield of production. And winner strategies, in our opinion, is to use all grape pomace through drying and grinding to obtain functional powder. We analyze the functional powder in terms of antioxidant activity, and now we are obtaining, and in this time now, we obtain a lot of good results concerning the in vitro activity of this powder. About the safety, of course, it depends from the starting material, but starting from biological production, we can obtain powder with a good granulometry, so size dimension, useful like that, with also the presence of anthocyanin can give very pleasant color of the preparation. And the first result was a small cosmetic line named Grape Skin Care, coming from winemaking industry byproduct. We are starting the in vivo efficacy test that very, very promising results in which we can see a very activity against the wrinkle. So activity is hydrated, is a lightening, efficacy, anti-age, and lenitive. Lenitive is very important because can reduce irritation after UV exposure or other chemical insult to the skin. So this is a preliminary test. The Grape Skin Care is a prototype of line. It's not yet in the market, but is very important to be a starting point for further study and for the valorization of the area, the geographic area. So I hope I stay in time. And I would like to thank you for your attention and let you free to make a question if you need.
Thank you very much, Dr. Paola. We will wait to see if we have some questions. Until the moment we don't have it, so okay. I would like to say that your presentation was very interesting, of course, because it valorizes waste, and this is a urgent need for industry and for the planet. And I would like to congratulate you in your work in this field. And also a curiosity, are you willing to test these products from the oils that you extracted that revealed promising results in other products, or will you stay in the hair field?
About oils or about the grape?
About the oils.
About the oils.
Yes.
I evaluate only one sources of waste.
Okay.
From nut, from this kind of nut coming from the same company that produce food and produce food, and then the residues from food production is coming in our laboratory for the test for activity in vivo in vitro. It could be very interesting to evaluate, to compare different suppliers, because it is very important to put some minimal requirement for ingredients coming from waste. This is the reason that I underline that the characterization and identity of the substance inside the ingredients can be very strictly investigated.
Okay, thank you, Dr. Paola. Now we have a question from Bruna Moreira. Could nanoformulations be the best way to introduce these compounds?
If we talk about oils, the problem for natural oils coming from waste is the same of oils. So it's greasy, it's a color, and sometimes smell. So we work some years ago also from residues from olive water, olive production, and it was very complicated for the smell and the color. The big problem are the color and the smell. So we have to use some pigment, blank pigment, white pigment to reduce the color, and you can use some natural oil, natural essential oil to change the smell and to make it more pleasure. About powder, it's a very chance to use it for skincare, but also for makeup. So you can have makeup with a function, a specific function, anti-age function, and so I think it's a good novelty for cosmetics, not only for texturizing agent, but also to obtain a real efficacy in the makeup.
Thank you very much, Dr. Paola. We will now move on to the oral communications session. And I would like to remember you that you can contribute once again with your questions in the YouTube live stream chat, so the speakers can answer them at the end of the presentations. And I would ask the speakers to stay until the end of the session so they can answer these questions. And also would like to mention that the book of abstracts and the diplomas for the participants will be sent by emails to the participants as soon as we have the ISBN number. And also that the book of abstracts will be available in our website too. So now let's move on to the first presentation. We have Anna Semik-Ojniak with the presentation Biomimetic Short Peptides in Medicine and Cosmetology.
Yes, that's something. Video doesn't work. I don't want to. Okay, so it should be. Okay, okay. So excuse me for delay. Half a minute. Yes, I want to say that I envy my previous speakers. They had a little bit more time than I have. So first of all, good morning to all participants. And first of all, thank you very much for this golden idea to organize this free online congress and giving me chance to present our achievements. My talk is divided in effect into two parts. In general, I will give you some information on biomimetic short peptides, and in effect, those will be the peptides and oligopeptides that can be used in medicine and cosmetology, that can be potential candidates both for cosmetic products and for and for drugs. So because of shortage of time, I think I omit the introduction, but then I give you some results on theoretical calculations. They have been done for two peptides, for dipeptides, and I present experimental fluorescence spectra, an assignment of theoretical structures to the issues that give this the fluorescence. And then I present also one slide on to prove that it is this sort of carrier peptide. And then I switch very briefly to tripeptide peptides, RGD analogs in mycobacterial infections, and the possibility to be used as both in cosmetic industry and in pharmacy. So all of us, we know that peptides are very interesting compounds. Peptides are short proteins in effect, and they play important roles in many biological processes like pigmentation, inflammation, cell proliferation and migration, angiogenesis, and extracellular matrix synthesis that is especially required for cosmetic products. The biocidal activity of natural peptides is effective against broad spectrum of bacterial, fungal, and viral pathogens, as well as parasites. We know here is the example of somatostatin, of peptide hormone comprised of 14 amino acids that is very important and has a great significant impact on neurotransmitters and cell proliferation. Also, I present just as an example the peptide hormone ghrelin, which is the hunger hormone, but we realized that natural peptides contained in drugs, they are isolated from the physiological secretion of animals or humans, and these processes are quite expensive. So in the time of developed biotechnological methods, in general medicine, in aesthetic medicine, and cosmetology, there is a great demand for synthetic peptides. And synthetic peptides are called biomimetics, and when they are in the cosmetic products, we may call these products cosmeceuticals. What are the benefits of biomimetics? No, so from the definition, they mimic the action of natural peptides, but because they consist of only several amino acids that in effect can be changed, can be altered, they easily penetrate the epidermis. They regulate the action of fibroblasts, improving appearance of the skin profile. They are also excellent humidifiers, since naturally they contain a structural water, what is useful in hydration in the cosmetics for hydration therapy. Moreover, they stimulate synthesis of natural collagen and elastin by stimulating the dermis cells, leading finally to skin regeneration and application in anti-aging therapies. Moreover, they present relatively high selectivity, efficient activity, low toxicity, low molecular weight, and their synthesis is cost effective. And if it happens that they are used in drugs, there is a possibility of oral administration. In general, the biomimetics we can divide into several classes. and signal peptides are capable of stimulating skin fibroblasts. Their action leads to an increased production of collagen and elastic fibers. And on the market, there is oligopeptide 68, such preparation. The carrier peptides are responsive for transporting and stabilizing elements such as copper and manganese. They simply complex these two metals. They carry them to the skin and allow the intake by epithelial cells. And on the market, there is the GHK-Cu3 peptides, glycine, histidyl, and lysine. And the third group, there are neurotransmitter inhibitor peptides, peptides that are capable of increasing minimal muscle activity. And first of all, they are applied in anti-aging cosmetics. So this is the summary of bioactive peptides currently marketed for inclusion as active ingredients in the skincare products. And we see here tripeptides, dipeptides, pentapeptides. We see many palmitoyl peptides, and also you see the copper mentioned here, GHK dipeptide. For organic chemists, it is not so difficult to get some sort of derivatives. And these derivatives, of course, aim to have different but still very useful properties. And
Dr. Anna, I'm sorry to interrupt. You have 1 minute, okay?
But it's impossible. So we took one heterocyclic representant quinoxaline that has many important biological properties. And we perform theoretical calculations, DFT and time-dependent DFT, and we got for such groups the quinoxaline substituted by a phenolic group and by pyridine and dipeptide, to which this scaffold was incorporated. And we followed the change of the structure, in the ground and excited state. We got this 2-dihedral angles, which were important. We derived many isomers of both of them, and we learned the spectroscopic properties and the barrier height, the barrier energy height, to have idea how they can interconvert one into another. And then to this substituted quinoxaline, the dipeptide was incorporated. And once again, we checked all these spectroscopic properties. It showed that they are different, of course. And then the excited state optimization by time, but the DFT method allowed us to get some information how the conformation changed, and we got the theoretical values of fluorescence maxima. And when it was necessary for us to assign appropriate isomer to appropriate fluorescence band, and so it was done. And we see that, for instance, in the fluorescence spectrum, one conformer, it was assigned to the open structure and another to the closed structure. And we have proven that these peptides easily chelate to copper ions, so we can classify them as the carrier peptides. And then I switch to the antimicrobial peptides, another group. The aim was to find a new drug against tuberculosis, which is very difficult to find because the Mycobacterium tuberculosis lives and proliferates in the leukocyte cells of human bacteriophage that in effect normally kills the bacteria after the phagocytic act. So what we have done, we have synthesized 3 groups of analogs of natural peptides, and we changed the sequence of amino acids, and we have got in each group at least one. For instance, here this oligopeptide gives important 60% antiphagocytic activity, and in the analogs of histamine, this is the plant hormone, also this code was repeated in the synthesized peptides. And also we found one with very significant antiphagocytic activity. And then finally 9 oligopeptides were synthesized, and in some of them we found really significant antiphagocytic activity. And in the conclusion, I may say that it is the results are very interesting, because more and more bacteria are antibiotic resistant, and so those are candidates, potential drug candidates in healing tuberculosis. And in the first case, this dipeptides presented, they can be used both in cosmetic industry, and because of the presence of quinoxalin that has many interesting biochemical properties, they can be used also as potential drug candidates. Thank you very much. Thank you for your attention. And it is very difficult to present in 6 minutes, so many slides. But nevertheless, thank you very much.
You're welcome. Thank you, Anna. Now we're moving on to cell suffrage, contributions towards the ecotoxicological evaluation of plant extracts and essential oils.
So, good morning. Can you hear me and see?
Yes.
Well, first of all, I would like to greet everyone and say thank you for the opportunity to present my work here. So essential oils, the essential oils market has been continuously growing for the past years, with Europe representing the biggest market share. These products are used in many industries, most of them in the food and beverages and the wellness area. And production is expected to continue growing, reaching 473 kilotons worldwide by 2027. So this demand by the industry has been accompanied with growing interest from the academia. Publications regarding essential oils and plant extracts have been continuously increasing for the past 20 years, with most of them focusing on development of new applications of these products. One thing that it's noticeable is that the research in the environmental field is still very low. In fact, the number of publications regarding environmental science is only 2% of the total published papers. And although plant-derived products are generally perceived as safe and healthy, there is a question that remains mostly unanswered: how safe these products really are to the environment. Let's take a look at the most industrially produced essential oils: orange, lemon, and mint essential oils. At the moment, there is no data regarding aquatic toxicity of these products. There is some data regarding major compounds found in these essential oils. For example, limonene is the usually the most abundant compound in orange and lemon essential oils, and it is reported to have toxic effects to Daphne magna at relatively low concentrations. The same for the mint essential oils. Usually the major compound is menthol, and it is also reported to have toxic effects to the fathead minnow fish. And as already said by Dr. Ana Oliveira as a little teaser in this morning presentation, the InnoVeb project aims to generate scientific data regarding essential oils and plant extracts of different plants in the Portuguese territory. These that can be used to develop new products. At the moment, extracts and essential oils of 5 plants have been studied for the ecotoxicological effects, namely the rock rose, thyme, basil, immortelle, and the vastly known hop plant. So the aquatic toxicity was assessed using the freshwater crustacean Daphne magna. This is one of the recommended test organisms by regulatory agencies such as the OECD or ISO to perform aquatic toxicity tests. And all tests were performed according to the OECD guideline 202. In brief, neonates are exposed to a growing set of concentrations, and the effect, in this case was immobilization, is observed and recorded after 24 and 48 hours of exposure. Then the results are statistically analyzed, and the EC50 value is determined. Moreover, a test validation with a reference toxicant is performed, and it was done using potassium dichromate. Looking at the results, the essential oils presented different toxicities. The essential oil from Immortelle showed no toxic effects up to 800 milligrams per liter. The rock rose essential oil showed some immobilization with a 48-hour EC50 of roughly 200 milligrams per liter. And by far, the most toxic essential oil was the one from thyme that presented a 48-hour EC50 of 11.20 milligrams per liter, which is low. Regarding the extracts, almost all the extracts presented no toxicity to Daphne magna, except from the chloroform extract from the flowers of hops that presented some toxic effects, although at very high concentrations. Essential oils and plant extracts can be classified for their toxicity to aquatic organisms following the globally harmonized system of classification and labeling of chemicals by the United Nations, which is also followed by the European Chemicals Agency. Of the extracts and oils that presented toxicity, only the thyme essential oil can be classified as toxic under the acute 3 category. To better understand this toxicity, and with data from our colleagues from the Plant Biotechnology Center of Beira Interior, they identified carvacrol as the most abundant compound in our essential oil, making up almost 80% of the total composition. Carvacrol is a phenolic monoterpenoid, and it is consistently reported as the major compound in essential oils obtained from this plant. And it is currently registered in the European Chemicals Agency as being toxic to aquatic organisms and aquatic life. And the high abundance of carvacrol in our essential oil can then be the explanation for the high toxicity observed. So, concluding, the essential oils from rock rose and immortelle present no risk to Daphne magna. All the studied extracts also present no risk. The essential oil from the thyme, Timbra capitata, has toxic effects to Daphne magna, and this could be explained by the high content of carvacrol present in the essential oils, and this compound is already reported to be toxic to aquatic organisms. In this case, precautions should be taken to avoid contamination of water bodies, especially by producers of these essential oils. Further toxicity assessment can be considered with other organisms to get a better understanding of the ecotoxicological effects. So finally, I have to give a special thank you to everyone involved in this product, and thank you very much.
Thank you, Celso. Now we are moving on to the next presentation, which will be given by Javier Alvarez, fatty acids composition, antioxidant, and antibacterial activity of ethanolic PLE extracts of 4 algae species from Galicia.
Thank you very much. Okay. Well, okay, I think this. Okay, my screen is flashing constantly. I don't understand why is this happening. Okay, is it now?
No, not yet.
Let's see.
Now we are seeing the presentation. Okay, it's okay.
You are seeing the presentation now?
Yes.
Okay. I will proceed then. I am Javier Chavez from the Nutrition and Bromatology Group from the University of Vigo, and I will proceed to present one of our latest works, which involved determining the fatty acid composition of 4 common macroalgae species, as well as characterizing the bioactivities, antioxidant and antimicrobial properties of the pressurized liquid extracts of the same species. Thus, the objectives of the study were to quantify the fatty acid content of these 4 common species, as well as to determine the feasibility of pressurized liquid extractions on these species from which potential bioactivities could be exerted. These 4 species were namely Ilmantalia elongata, Ulva intestinalis, Ulva lactuca, and Phystoceira baccata, which were collected from the Ferrol Ria in Galicia, northwest of Spain. The choice of these 4 species was considered being widely distributed, especially in the European Atlantic shores. They are accounted as well for rapid and annual growth, which means that they would have a highly available biomass that could be therefore employed as a feasible source of potential alternative cosmetic ingredients. Also, it should be considered that they are, especially sea spaghetti, Mantalia elongata, gadwit, Ulva intestinalis, are edible and used in cuisine. Therefore, we extracted and purified the lipid content of these algae using the Fogg method. And once these purified lipids were extracted, they were derivatized to fatty acid methyl esters, which were then subjected to identification and quantification using mass spectrometry coupled with gas chromatography in the conditions, with the set of conditions that are shown. We observed that the highest fatty acid content was present in Phystoceira baccata. As it is already reported in scientific literature, macroalgae are not a very good source of fatty acids. Yet, Phystoceira baccata displays relatively high concentrations of these bioactive lipids, of which are somehow remarkable the concentrations of palmitic, myristic, palmitoleic, stearic, and oleic acids. Also, it is worth mentioning that only Phystoceira baccata seem to contain albeit at very low concentrations, arachidonic and eicosapentaenoic acids, which are recognized as highly bioactive fatty acids. Simultaneously, we also, from the same species and the same samples, we subjected them to pressurized liquid extractions using pure ethanol as solvent. The purpose of this was to extract the greatest amount of lipophilic molecules that could be present in this species. The rest of the parameters were set up based on already optimized methods. Hence, as mentioned, we tested the antioxidant and antimicrobial activities that could display this algae. Using a DPPH radical capture assay, we determined that Ilmantalia elongata and Phystoceira baccata displayed significant capture of these radicals. Specifically, Ilmantalia elongata was able to inhibit the release of free radicals at 64 micrograms per milliliter, and Phystoceira baccata much more lower concentrations at 28 micrograms per milliliter. It is worth mentioning that for the Ulva species, we could not observe a significant inhibition of this radical liberation, which could be related to their low content in lipophilic substances that could be also be argued to be due to using ethanol as solvent. For the antimicrobial assay, we employed two model species for gram-positive and gram-negative bacteria, namely Staphylococcus aureus and Escherichia coli. These were cultured in microplate at different concentrations in the range that are presented, and differences in optical density were measured by spectrophotometry. From the four species, all the four species, exert some degree of inhibition on the bacterial growth, yet only Phystoceira baccata shows significant inhibition on this same bacterial growth. Specifically, we observed an inhibition of more than 40% of inhibition for Staphylococcus aureus and more than 60% for Escherichia coli. Thus, it should be recognized that in relation to the low inhibitions shown for the previous species, this could be related to the low concentration of the extract used for the assay, as well as being related to the relatively high content in fatty acids that was accounted for Phystoceira baccata. In conclusions, from the former macroalgae species study, Phystoceira baccata accounts for the greater fatty acid levels, and Phystoceira, as well as Ilmantalia elongata, ethanolic pressurized liquid extracts, display significant antioxidant properties. As well, Phystoceira baccata was the only species to show significant antibacterial activity at these low concentrations. All these results would conclude in would conclude in assessing this species and this technique of extraction as a feasible method for including this species as potential and alternative sources of cosmetic ingredients. That will be all. Thank you all for your attention.
Thank you for your presentation. It was very interesting. And now we will listen to Paula Plasencia, bioactive properties of different extracts obtained from the aerial parts of blueberry and raspberry raw materials. Paula, are you ready?
Are you seeing my presentation?
Yes, we are seeing it.
I choose it. Okay. Well. Good morning. My name is Paula Plasencia, and I'm presenting some results of my work obtained under my doctoral program. This work is focused on upgrading the commercial potential of raspberry and blueberry plant byproducts, which are typically discarded, presenting no economic value. The outline will be sample preparation and extraction process, evaluation of extracts antioxidant activity, evaluation of extracts antimicrobial activity, and conclusions. Plant samples were obtained from a private company interested in exploring the bioresidues of raspberry and blueberry plants as sources of added value molecules. After they dried, the raspberry and blueberry aerial parts were grounded. And here we have the plants before and after being grounded. On the left, raspberry samples, on the right, blueberry samples. The extracts were obtained using ring methodologies, infusion, decoction, maceration, and ultrasound-assisted extraction, being the latter two extraction performed with an 80% ethanol water mixture. The 4 tubes on the left refer to blueberry extracts, and the 4 tubes on the right refer to raspberry extracts. The aqueous extraction process followed this diagram: decoction or infusion, filtration, and lyophilization. The hydroethanolic extraction process followed this one: maceration or ultrasound-assisted extraction, filtration, ethanol evaporation, and lyophilization. Samples extracts were named according to Table 1. Extracts were studied regarding their antioxidant properties using the oxidative hemolysis inhibition assay, and antimicrobial properties were studied through microdilution method against pathogenic bacteria. For the antioxidant evaluation, we used erythrocyte solution, a microplate reader at 690 nanometers every 10 minutes. The results were presented in IC50 at 60 and 120 minutes in microgram per milliliter, and the positive control was Trolox. For the antimicrobial evaluation, extracts were incubated, diluted, inoculated with gram-positive bacteria, Enterococcus faecalis, Listeria monocytogenes, and Staphylococcus aureus, and gram-negative bacteria, Escherichia coli, Klebsiella pneumonia, Morganella morgani, Proteus mirabilis, and Pseudomonas aeruginosa. After inoculation, they were incubated for 24 and 48 hours at 37 degrees. INT was added, and 30 minutes more at 37 degrees, and finally our results were analyzed. Antioxidant activity results. Both raspberry and blueberry samples extracts exhibited a similar activity, with raspberry decoction and blueberry decoction extracts presenting better results with 21.9 and 20 microgram per milliliter. These extracts presented similar activity, and one of them even higher than the one of Trolox control with 21.8 microgram per milliliter. For the antimicrobial results, all the extracts presented inhibition capacity against most of the tested bacterial strains, being the ultrasound-assisted extracts the most active ones. Raspberry samples showed the highest activity, inhibiting the gram-negative bacteria Escherichia coli, Klebsiella pneumonia, Morganella morgani, and Proteus mirabilis with a minimum inhibitory concentration of 5 milligram per milliliter. In contrast, to inhibit this bacteria, a concentration of 10 milligram per milliliter of blueberry extract was needed. Regarding the gram-positive bacteria, raspberry extract presented a minimum inhibitory concentration of 2.5 milligram per milliliter against Enterococcus faecalis and methicillin-resistant Staphylococcus aureus. For blueberry extract, a minimum inhibitory concentration of 10 mg per milliliter was determined. In conclusions, these results give us raspberry and blueberry byproducts as novel candidates for industrial application. The best extracts may be used in cosmetic formulation, upgrading those bioresidues. Next step in this work will be to test and to validate these capabilities in cosmetic formulations. Thank you for your attention, and I hope I was successful in explaining the results of the topic under discussion.
Thank you very much for your brilliant presentation. And now we will give the word to Sara Gonçalves with the evaluation of cosmetic properties of natural ingredients in the Trás-os-Montes area, a PhD project.
So I hope everybody can hear and see me.
Yes.
Okay. So I would like to thank all of you to be present here today. I would like to thank the scientific committee for choosing my work to be presented here today. It's a PhD project. It lacks results. However, they will be available along this year. It's an evaluation of cosmetic properties of natural ingredients that are common here in the Trás-os-Montes area. And as you may look upon my first page, you may start guessing which natural ingredients will be used, but more of that ahead. So firstly, we had to access what do we know about this theme, namely, what is a cosmetic? And we found a definition by the Council of the European Communities that established the laws of the member states relating to cosmetic properties, cosmetic products, that says that it's a product that's applied to the skin, to the body, with the purpose of beautifying, cleansing, or improving and enhancing attractive features. So it's everything that's a soap, a shampoo, a toothpaste, makeup, everything we use on a daily basis. And we also found out that cosmetics was used throughout the history. There are many more evidence than the ones presented here, and I'm just going to highlight one. It's from the Middle Ages. It's Queen Elizabeth I. She used Venetian ceruse. Ceruse is a mixture of vinegar and white lead, and that would give her that white purity look that she was known for. She was quite the trendsetter back then. However, ceruse was very, very toxic to the skin. So she would apply more ceruse to cover up the damage. That would damage even more the skin, and so she would apply even more ceruse. Although in ancient times they used cosmetic with toxic properties, today it is not much more different. A study performed by the Danish Council Think Chemicals found 65 chemicals of concern in 39 products. And 3 products contain illegal ingredients in the EU, namely this mascara that is sold specific in Spain and Sweden. The natural cosmetic market has also grown since the consumer took consciousness of the concept of natural-based ingredients. A study performed in France showed that the sales went up for natural cosmetics. 80% of the French women already have decided to buy natural or organic products, and 50% of the consumers decided to buy these products when they realized the ecological impact of a non-organic product. We also know that this is a market that is constantly growing. Analysts anticipate a global revenue to reach up to $54 million by 2027. Thus, the use of natural and organic cosmetics becomes increasingly important, and this requires a strong investigation into benefits that fruits and plants can bring to our health. So we establish 5 aims with all this knowledge that we have assessed. Firstly, we want to, after selecting the natural ingredients, we want to assess their potential to promote longevity, prolificity, and antigenotoxicity. Also, the potential to promote DNA damage. The natural ingredient with the best results in these tests will be used to perform a chemical characterization and also to evaluate genoprotection in human lymphocytes. Finally, the ingredient will be evaluated if it can be incorporated in a cosmetic. So we selected 4 ingredients that are common here in Trás-os-Montes. Elderberry flower and berries. Elderberry is a wild tree. Almonds and olives that come from organic farming, and grapes that only will be harvested September this year. These last 3, almonds, olives, and grapes, come from a non-intensive agriculture. For our first aim to access if the natural ingredient has the potential to promote longevity, prolificacy, and antigenotoxicity, we'll be using a SMART test. SMART test uses genetic markers phenotypically observed in adult tissues, and it detects mitotic recombination. For the other test, to promote DNA damage repair, we'll be using commercial cell lines, and we'll be using the COMET assay. COMET assay can measure single and double stand breaks as also AP sites. So with all these tests, we can determine which ingredient has the best results, and that will be used to perform a phytochemical composition and also an antigenotoxic potential in human lymphocytes using also the COMET assay. Finally, for the last aim, to analyze which natural ingredient can be incorporated in a cosmetic and so benefit from its antigenotoxic properties. Studies have shown that antigenotoxic properties are related with anti-aging properties. We are currently discussing it with a corporation that is known for formulating natural and cosmetic products, and also for selling them. However, I cannot disclose any more than this. Maybe next year I have some more news for you. And I'm going to just pass right through my schedule throughout the PhD. I want to thank you all for your attention. I want to thank also SECAF for funding this research, and of course, my supervisor, Professor Isabel Gaivão.
Thank you very much, Sara, for your presentation. And I wish you the best luck for the project, okay? It seems very interesting. And now we will move on to the last presentation with Silena Alves. Humulus lupulus, cosmetic application of extracts obtained from cones and vegetative parts. Silena, whenever you want.
Okay. Hi, good morning, everyone. My name is Silena Alves, and I will present my work entitled Humulus lupulus, cosmetic application of extracts obtained from cones and vegetative parts. Hop is known worldwide as an essential raw material for the beer industry, and recent studies have been revealing several plant health benefits in the beer production process. Corn of hop are used and the vegetative part, such as leaf and stem of the plant, are discarded. However, the literature indicates to have a quite interesting property. Concerning the chemical composition, hop is composed by beta acid, alpha acid, polyphenol, essential oils, and nutritional components. As I said, hop plant has important pharmacological and biological properties, such as antioxidant, sedative, antibacterial, anti-inflammatory, estrogenic, among others. The aim of our study was study the phytochemical profile of polar extras of hop and of the essential oil of Thymus digis, and further evaluated the antibacterial and antifungal capacity. Also, a gel formulation were developed by incorporation of different concentration of hydroalcoholic ester of cones and vegetative part of the nuggets and polaris variety and of a spontaneous hop sample. In addition to that, the stability of the different formulation was evaluated. For that, a hydroalcoholic extraction of cone and vegetative part of hop was carried out to phytochemical profile analysis, evaluation of antibacterial and antifungal activity, preparation of formulation contained hop extract. It was also done the obtain of essential oil from Thymus digis and the antibacterial and antifungal capacity evaluated. The determination of the phenolic profile was done by HPLC-MES. The evaluation of antimicrobial activity were performed by microdeletion broad method and the diffusion in agar test. Additionally, the stability of the gel formulation was evaluated through a series of physical chemical tests, such as pH, organoleptic analysis, texture, light, relative humidity, mechanical vibration, centrifugation, and density. In our study, 30 compounds were identified, 5 and alpha and beta acid, 10 phenolic acid, and 15 flavonoid. Generally, extracts obtained by cones were the richest in phenolic compounds. Concerning of alpha and beta acid compounds, we were identified the comulpon, comulon, umulon, colupulon, adreomulon, and lupulon compounds. These compounds represent the major in cones extracts. Glycoside form of the flavonoids camphorol, quercetin, and criosterol were identified in vegetative part of whole extracts. Xanthomol, prenulated chalconoid, is present in cones as described in literature, and in the present study was found only in the nuggets variety. Flavonoids were mostly found in spontaneous whole in the vegetative part of the plant. In the preliminary stability test, the formulation showed no change in the organoleptic analysis, mechanical vibration, and in the centrifugation test, presenting acid pH with an apparent density of 1.08 to 1.14. In the accelerated stability test, the formulation showed no change in the extreme consecutive temperature variation and in the relative humidity. In the light test, the formulation showed degradation. For texture size, the consistency and the cohesive of the formulation increase comparing the gel-based formulation to the increase in the concentration of extract. In spectrophoton test, the formulation with hydroalcoholic esters from plants can showed greater absorption, which may be related to the chemical hindrance in alpha and beta acid. Old hope extract had no antimicrobial effect at the tested concentration. The essential oil of Thymus zigis had showed antibacterial and antifungal activity against all strains of bacteria and fungi tested with rigor potential against Staphylococcus aureus than for Pseudomonas aeruginosa. As conclusion, the stability test did not reveal signs of separation of liquation of the preparation gel sample. It's important highlighted that the present study explored the vegetative part of hop that remain poorly studied, allowing propose them as integration in cosmetic area. In this slide, these are some references that use in our work. To finish, I would like to thank IPV and Research Center for financial support, and a special thanks for your attention.
Thank you, Silena. And now I will ask all the speakers to answer the questions that the audience has been asking. So the first question goes to Celso Ferraz. It comes from Sara Gonçalves. Celso, okay. Did you extract the essential oil yourself? If yes, which process did you use? And would there be a difference between self-extracted and commercial essential oil in terms of toxicity?
Yes, thank you, Sarah, for the question. I did not extract the essential oils myself. That was done by our colleagues in the Polytechnic of Castelo Branco. So I don't have that information on which process was used. Regarding the differences, yes, there can be a difference. especially if you collect wild plants to obtain an essential oil. The composition can vary from where the plant is collected, the time of year when it is collected. There are many environmental factors that can affect the ability of a plant to produce essential oils. Regarding commercially available, well, usually a commercially available essential oil has to have a specific composition, or at least a range of composition of the main compounds. So yes, the toxicity can vary according to the form or how the plant is collected, and also the extraction process can affect the composition of the essential oil.
Thank you, Celso. And now the next question will be to Javier from Teresa Silva Braz. Okay, regarding the pressurized liquid extraction, the ethanolic extract, did you quantify the phenolic fraction? And also, did you correlate the activities achieved with the presence or absence of some compounds?
Thank you for the question. And it's actually a very good question. We did not analyze for the moment the phenolic compounds present. And considering that using ethanol as a solvent, as many of you may be aware, it absorbs the lipophilic fraction. So this means it will absorb as well as fatty acids, also, for example, carotenoids. Which many of them, like for example, fucoxanthin or beta-carotene, are considered phenolic compounds. Thus, we aimed to correlate the exerted bioactivities and properties with the supposed level of these compounds. But we did not determine the next step to make, but no, not for the moment. I hope I'm getting a message that my internet connection is low. Well, I hope you have heard me to successfully answer your question.
Yes, Javier, in fact, it did fail, but I think it was possible to understand. Yes, thank you. And now the last question will be to Paula Plasencia from Teresa Silva Braz. And the question is, did you identify some group of compounds for the different extraction yields? And if yes, what were the differences in terms of extraction yields?
I'm sorry, can you repeat the question, please?
Yes, Paul, of course. Did you identify some group of compounds from the different extraction yields? And if yes, what were the differences in terms of extraction yields?
Okay, thank you very much for the question. We have identified some phenolic compounds by HPLC DADMES. We found some flavonols and phenolic acids, and in some cases some tannins. The yields, well, we didn't determine the yield yet because we have to decide further on work what extracts to be used, and then in that time we are going to determine also the yield of the extraction.
Okay, thank you very much, Paula. I think we have no more questions. So I want to thank you all for your attendance. And now we have 10 minutes to take a brief break and to see the posters at the corresponding links. And then we will move on to the last pitch session, which will be moderated by my colleague, Dr. Cristina Caleja. Thank you again. Hope you're enjoying the conference.
Good morning and welcome again to the first online Congress of Natural Products Application, Health, Cosmetic and Food. We remind all the participants that the posters are still available online and distributed by areas, and with the possibility that you can download all the information in PDF format. Let's start the last speech section in the area of cosmetics. I remind all the participants who follow us on the YouTube channel that they will be able to leave their question in the channel chat during this section. The organization will select some questions that will be asked to the speakers at the end of the section. For a matter of time, and if it is not possible to explain all the questions, they will be sent particularly to the speakers to be answered later. I remember the speakers that this section is a continuous section of a short lecture lasting only 3 minutes each. To avoid delays in the program, please do not exceed the proposed time limit. The questions proposed by the channel participants, as I have already mentioned, will be exposed at the end of the 6th speech. So we will start. I move on to the speaker, Ana Costa, to start her pitch, please.
Can you hear me?
Yes.
Good morning to you all. My name is Ana Costa. I'm from University of Lisbon. And today my pitch presentation is about the development of formulation that convert garbage into glamour. And we are interested in assessing the organoleptic properties of formulation containing tomato extracts from residues. The aim of my project consists in collect tomato waste from a tomato Portuguese company. Then we use the green extraction methods to obtain lycopene enriched extract that are being incorporated into cosmetic products.
Sorry, Anna, you have a presentation.
Sorry?
You have a presentation.
Ah, I forgot to put.
You have to share the screen, please.
Yes, sorry. Are you seeing now?
Yes, we are.
Should I start from the beginning?
Yes.
Okay. My pitch presentation is about the conversion of development of products that convert garbage into glamour. And we are now assessing the organoleptic properties of formulations containing lycopene enriched extracts from tomato residues. The aim of my project consists in collect tomato waste. Then we use green extraction methods to obtain a lycopene enriched extract that will be incorporated in cosmetic products. Here is a picture of 2 microemulsions with and without the tomato extract. The next step was to perform a sensorial analysis in order to know the opinion of consumers. And one of the aspects that we asked was about the odor intensity and odor perception. And for both formulations, the odor was classified as slightly perceptive and undefined odor. Then we asked about the probability of buying different care products with the odor of microemulsions, in particular a face cream, a body cream, and a shampoo. And we observed that the majority of volunteers prefer the formulations with the tomato extract. But when we ask about probability of buying the toothpaste with the odor of both microemulsion, the probability was really low. The next step was to ask about the color. And the color perception for both formulation was also classified as a pleasant color. Then we ask again the probability of buying care products with the color of these formulations. And we observe that the majority of volunteers prefer a face cream, a body cream, and a shampoo with the color of formulations without the extract. But when we informed volunteers that this formulation contained a tomato extract with antioxidant probabilities, we observed an increase in the number in the probability of buying these care products. Once again, the color of this formulation was not suitable for development of toothpaste, even when we informed the volunteers about the property, about the composition of the formulations. As a key point, to achieve a successful marketing product, we need to think about the type of the product, the composition of the final product, the source of bioactive agent, as well as the biological and pharmacological properties. And thank you all for your attention.
Thank you, Ana Costa. We will move on the next speaker, Ana Rita Silva.
Morning, everyone. Today I will be talking about part of my PhD work, and I will be presenting the work entitled The Optimization of a Tannin-Rich Extract using RSM. Cytinus hypocystis is a parasitic plant with a great potential to be applied in the cosmetic industry, since in our previous work, we correlate its high tannin content with its excellent cosmeceutical properties. Thus, the objective of this work was to optimize tannin concentration in the extract using heat and ultrasound-assisted extraction. We applied RSM to find the optimum combination between time, temperature or ultrasonic power, and solvent ratio to maximize the concentration of tannins in the final extract. RSM allows us to optimize processes using a small number of experimental runs. In this case, we applied a rotatable central composite design, which represents the 5 possible values for each one of the 3 independent variables. These natural values were converted to coded values that were later combined in these 20 experimental runs. We performed the runs in the lab and we obtained the final concentration of tannins for each method. A polynomial equation was fitted to the experimental values obtained in the lab and allowed us to access the linear, interactive, and quadratic effect of the independent variables on the final response. On the right, we have the polynomial equation for heat-assisted extraction, which allowed us to construct the following 3D representations. As we can observe, the solvent percentage, the linear effect of solvent percentage had the highest positive impact on the final response. As we can see in graph 2, independently of time, the tannin concentration was maximized when the solvent percentage was close to 100. Similarly, in graph 3, at lower temperatures, the response was maximized when the percentage was close to 100. Regarding ultrasound-assisted extraction, once again, the linear effect of solvent had the highest impact on the final response. As we can observe in graph 2 and 3, independent of time or ultrasonic power, we had the maximum response in solvent percentages close to 100. So considering all response variables, the optimum global conditions were predicted. Heat-assisted offered the highest concentration of tannins. However, ultrasound offers sustainable and economic advantages in time. Our next step will be to establish a correlation between the optimum conditions and their respective bioactivities. Thank you for your time and attention.
Thank you, Ana Rita, for your presentation. And now we will move on to the next speaker, Bruno Melgar Castaneda. You can start your presentation, Bruno.
Okay, thank you, Cristina. Okay, just give me a second. It's now? Okay, so good morning, everyone. My name is Bruno Melgar Castaneda, researcher from the Monta Research Center, and the topic I will be presenting today deals with the ultrasound extraction and time effect. What's going on here? Just give me a a second. Sorry. Oh my God, I'm sorry for that. Okay, I'm sorry for that. I would like to start by the end of optimization presentation, like the one we saw before, showing some two-dimensional graphs and stuff, and the terminology usually employed or encrypted code for others, things like design of experiments, central composite design, or other now world famous response surface methodology. So if experiment design does not look familiar to you, just allow it a couple of months, because according to this data from the Web of Knowledge using the RSM, the number of publications is growing rapidly, and great numbers in the biotechnology area like ours. If we look closer, we will also be able to identify some specific trend, like the uses of optimization with ultrasound-assisted extraction coupled to response-based methodologies, and even more complex modeling and prediction approaches like artificial neural networks. But okay, before we continue, it's a good point to remember the basics. Usually for laboratorial experimentation, we gather some vegetative matrices, we sample prep, and then in this particular case, we use cavitation effect of ultrasound, which compress and expand the nucleated bubbles, which apply dissimilar pressure to the vegetative cell and result in the release of their inner bioactive compounds, the one we use for the natural applications. So from here, we could go back to the fancy and complicated terminology, or we could focus on easier graphs, but also meaningful data. For instance, in this work from our group, before we started the optimization process, we decided to focus on some important but yet overlooked information, like the effect of time. While using ultrasound, we explore in 5 different species with very short ranges of time, from 10 seconds until 12 minutes. And although we found significant differences, we could not observe any specific trend like more time, more extraction. We have noticed some publication with very long times, like 52 minutes or 30 minutes. And even in my work in here, that is 2.5 minutes as an optimum point of extraction. But the important thing in here is not how many complex models we can use, or hyper long or shorter times are better. The important thing is to keep asking ourselves, does my results make sense? Will 15 minutes ultrasound extraction properly optimize my bioactive extraction process? I mean, in optimization experiments, polarizing responses is not always the answer. We have to be aware of the future processes cost, not only from the economic point of view, but from the environmental one as well. So my personal suggestion is that academia has to be aware of industrial challenges and limitations, and vice versa. But always as a scientist, we have to keep it real in order to make it work. So understanding, modeling, and processing data are magnificent tools, which should always be aligned with critical thinking and decision making. That's it. Thank you.
Thank you, Bruno Melgar. We now advance to the fourth pitch. We will move on to Diana Andrea Tavares Pinto. Diana, you can start, please.
Okay, thank you. Good morning. Can you see my presentation?
Yes, we can see.
So good morning, everyone. My name is Diana Pinto, and I'm a PhD student from Requinte Polytechnic of Porto. Today, I will present my recent work that focuses on the valorization of Castanha sativa shells as a potentially valuable raw material for cosmetic industry. So chestnut shells are an abundant and underexploited byproduct of chestnut processing industry, mostly reused as fuel and as a source of tannins in wine aging process. However, large amounts of this byproduct are still available. And for this reason, ongoing studies have focused on searching for novel applications, highlighting that these shells are excellent sources of bioactive compounds particularly polyphenols and vitamin E, and reinforcing their potential of valorization as natural antioxidants. It is also worth to underline that the implementation of eco-friendly extraction processes embraces a huge challenge for industry. So the main aims of our study was to employ the ultrasound-assisted extraction to the recovery of polyphenols from chestnut shells and to provide a preliminary evaluation of their cosmetic potential. For this, the shells, after dried and ground, were submitted to an ultrasound-assisted extraction at 70 Celsius degrees for 40 minutes, and the aqueous extract obtained was further characterized regarding total phenolic content, antioxidant and anti-radical activities, skin cell viability, phenolic composition, and finally, ex vivo skin permeation using France diffusion cells. So the results revealed a high phenolic content and strong antioxidant and anti-radical properties based on the IC50 values ranging from 32 to 65 micrograms per milliliter of extract, and the highest radical scavenging efficiencies were obtained against nitric oxide radical and hypochlorous acid. Regarding the phenolic composition, the major phenolic compounds were ellagic acid, a caffeic acid derivative, and epigallocatechin. The results also proved that the keratinocytes and fibroblasts viability did not decrease significantly up to a concentration of 100 micrograms per milliliter of extract. In addition, the skin permeation study showed that ellagic acid and epicatechin were able to permeate the human skin after 8 hours, displaying higher levels of ellagic acid. In conclusion, these findings suggest that the chestnut shell extract obtained by ultrasound-assisted extraction have some efficacy and safety, pointing out a promising application in cosmetic field. Finally, I would like to thank the national and international funds that supported this research, and thank you all for your attention.
Okay, thank you, Diana Pinto. It's time to move on to the next speaker, Maria Aurora Soares da Silva. Hello, Maria. You can start your presentation.
Olá, bom dia. Good morning. Thank you for the opportunity of presenting a part of my work concerning bioactive properties of 6 algae from the Iberic Peninsula Sea. Algae are known to be a good source of bioactive molecules described as having antioxidants, antimicrobial properties, among others, with a potential application in food, medicine, and cosmetics. In this work, the antioxidant profile, antimicrobial capacity, and neuroprotective properties were studied. The alga under study was Ascorphilium notosum, Bifurcaria bifurcata, Fucus spiralis, Imantalia elongata, Sargassum muticum, and Iondaria pinitifida. The extraction were made by microwave-assisted extraction in the optimized conditions described in this slide, and the microorganisms used to the antimicrobial tests are also presented here. Some experimental notes, we did the antioxidant profile. Antibiograms are made by plate diffusion method. All the extracts were brought to the concentration of 20 milligrams by milliliter, and we did enzymatic tests based in the Hellman's method. The main results of the antioxidants and antibiogram tests are presented in this slide. It's notable that all algae species tested have effect against Staphylococcus aureus and none against the E. coli strain. In this slide, we can see the acetylcholinesterase and butyrylcholinesterase inhibition rates. Main conclusions. The most expressive antioxidant profile belongs to Ascorphilium nodosum. All algae extracts presented capacity to inhibit the Staphylococcus aureus growth, and the Bifurcaria crude extracts was able to inhibit all microorganisms tested except 2 E. coli. Bifurcaria bifurcata presents the best neuroprotective potential, being able to inhibit the butyrylcholinesterase action. Thank you for your attention.
Okay, thank you for your presentation, Maria Silva. I remind all the participants who follow us on the YouTube channel that continue to post their questions in the chat to be exposed to the speakers. So we advance to the last speech. We will move on to Patricia Brunziska. Patricia, you can start.
Okay, thank you. Is it okay now?
Yes, it's okay.
Okay, good morning. My name is Patricia Brudzinska, and my pitch presentation is about application of plant-derived colorants in cosmetic products. Nowadays, we observe a rebirth of the application of natural materials also in cosmetic industry. There is a strong need among consumers and companies to create such a cosmetics. There is a wide spectrum of plant-derived ingredients that are applied, but there's also a group of materials that is essential, particularly in color cosmetics. They are dyes. Nowadays, mainly synthetic or mineral dyes are used, but in the past, dyes were obtained mainly from different plants. But for some time, they got replaced by cheaper, easier to obtain, but not so beneficial for the environment and our health synthetic ones. But there is a huge selection of colorants in nature. There's a large number of fruits or vegetables that can be a source of color, like anthocyanins, carotenoids, betalains, or quinones. Thanks to the content of polyphenols and other biological substances, natural plant colorants can have other properties in cosmetics like antioxidant or antimicrobial, but their stability under the influence of light, high temperature, or pH may not be satisfactory in cosmetic application. Dyes are soluble in medium and can be used to dye cosmetic formulation, but to change the skin color, the pigments are needed. So to obtain plant-derived colorants that are insoluble and simultaneously to improve their stability, we've got different methods like obtaining polymeric dyes or laking natural dyes onto an inorganic substrate. And in the experimental part, materials that were used were plant extracts like strawberry extract, togberry, blackcurrant, black lilac, watermelon extracts, or cocoa or turmeric. And of course, inorganic substrate like talc and hydroxyapatite. Obtaining compounds were examined by skin coloring method to check whether they are able to color the skin and to what extent. To check their chemical structure, infrared spectroscopy was made, and to verify their stability under the influence of light. Samples have been treated by UV radiation, and to check their stability under the influence of temperature, they were placed in the incubator. So the results, intensity and stability of color were varied because of different amount of absorbed colorants onto substrates and because of different content of coloring substances. But some of tested plant extracts can have a potential to be applied in cosmetics, but it still requires further research. But maybe they can be an alternative source of color in cosmetic industry. And thank you for the attention, and thank you, committee, for possibility to present.
Okay, thank you, Patricia, for your presentation, and thanks to all the speakers for their interesting pitch. And now we will start the questions section for the participants who accompany us through the YouTube channel. Let me check if we have questions. The first question will be to Bruno Melgar from Márcio Carocho.
Yeah.
What do you propose to bring academy and industry closer?
Basically, what I was trying to show with the work is that sometimes we publish a lot of papers when we think that an optimized process is done, and we are considering like really long time of extraction, that when we do it, when we translate it to the industry, it won't be possible, or it won't be real, or it will be so expensive. So basically what I'm trying to say is we have to focus not only in the important methodologies that they have like really good look graphs, but we have to think further how will this process will be implemented correctly, and if that is really going to work.
Okay, thank you, Bruno. The next question will be to Diana Pinto from Márcio Carocho too. Is it preview to be in the market anytime soon?
The extract, for now, not yet. But we are performing more assays about the efficacy and safety. We will perform assays using in vitro models, 3D models, like EpiSkin, to know about the skin irritability. And we will do a patch test to know the effects for in vivo application.
Okay, thank you, Diana. The next question will be able to Diana Pinto too, from Teresa Silva Braz. What was the skin model you used to perform the France diffusion cell assay?
We use I think the participant is asking about the skin model. I use a human skin recovered from abdominal surgery. That we have a protocol with a hospital, and a surgeon provides us this human skin recovered from surgeries.
Okay, thank you very much.
Thank you.
And the last question for Patricia. It's a curiosity. For how long did the colorant last in the terms of shelf life?
Could you hold the question one more time, because I hadn't heard it.
For how long did the colorants last in terms of shelf life?
The shelf life is still in the experiment, so we are still making experiments. So now from this point, I can't answer this question because it's still in progress.
Okay, thank you, Patricia, and thanks again to the speakers and also to the channel participants. We move now to the keynote, and it's an honor for me to present Maria Isabel Monteiro from Colab. In 2009, Isabel Monteiro starts her professional path in the cosmetics and cleaning products formulation area, and since 2013, she works at Colab Portugal. First as a new product implementation, and then as a blending specialist at CPD technical department. In 2018, she joined the R&D team as a product developer, and she continued her academic studies, taking her master's in pharmaceutical technology in the Faculty of Pharmacy at the University of Porto. At this moment, Isabel is attending the first year of PhD in Sustainable Chemistry at the Faculty of Science in the University of Porto, where she graduated in chemistry. Isabel, thank you very much for your presence at the Congress. I remind you that the presentation will have a duration of 25 minutes and that the participants you'll once again be able to ask their question through this chat on the YouTube channel, and then we put to Isabel at the end. Isabel, you can start your presentation, please.
Hello, good morning or good afternoon to all of you. You are hearing me now?
Yes. Yes,
okay.
it's okay.
So I will start presenting. Okay. Please let me know you are seeing my presentation?
It's okay, yeah.
Yes, it's okay.
Okay, okay, thank you. So first of all, I want to thank you for the opportunity to be here and presenting here. What I'm talking about is what all the speakers are talking about is the natural cosmetic products and the trend. It should be a trend or started with a trend. So I will give my point of view as a product developer. First of all, let us see how this is going on. So I will start with the collab presentation is where I'm working now as a product developer in R&D, just to show you or for you to know this company. Now, then we will, I will start to talk about the natural, is a trend or a new reality. What is driven this is the consumer expectations, their demands, what we as a product developers should do, and what are our challenges. And also take this opportunity to show some of our innovations here at Collab. So Collab has more than 50 years. It started in 1965 with tinplate cans for cookies, and then started to produce aerosols. It's hard to explain it so short of time, the history of this company. Just to point out two things important for this presentation. In 2006, it was launched the Innovation Center, and the beginning of the R&D where I'm working now. And this is a multinational company that was born here at Alcâmbra. It's a Portuguese multinational. It belongs to RAR Group. And is truly all over the place, all over the world. When in 2013, it started a strategic alliance with the One Asia Network. So in the next slide, you can see that we are all over the world. We are in Portugal, Spain, Poland, Brazil, Mexico. One Asia Network, we have presence in Australia, China, India, Japan, Thailand. So we are a global company. For this reason, we have a network of 18 manufacturing sites, mainly focused on aerosol filling here in Alcâmbra, Kleszów in Poland, Bad Schmiedeberg, Germany. We have also liquids filling, packaging solutions, metal packaging, plastic packaging, co-packing. So we have a complete solution to offer to our customers. Our focus is in a beauty and personal care, health care, home care. mainly packaging offer in tin-plate aerosols, general lines for cans, food, paints, plastic packaging also. Here we have also, it's relative new, the IPS, comparing to the story of life of the company. It's an innovative product solution where we can offer to our customers products ready to sell with already stability tested, pre-industrialization validation, claims. So we can offer these products to our customers. These are developed together with the R&D. So the IPS will sell this portfolio products. So our main work is mainly in the cosmetic area, in the liquids, aerosol. We develop new products, we optimize the existing ones, we propose improvements in the cost, claims, substantiation, technical support. We are always connected to universities. We are always increasing our knowledge, having in mind that we need always to offer different things and to be faster in development. The R&D is divided in 4 countries here in Portugal, Poland, Germany, and Mexico. Now you know a little bit about Collab. Now let us see how the natural cosmetic is a trend or will be a new reality. Maybe it started like a trend. Like said before, the natural is connected to something that is safe, is good for you. Maybe it's go in line with is existing with the food industry, because if you can eat, it's safe for you and you can use it as a cosmetic. Consumers perceiving the message like this. So the natural is the start of a new concept, new trends, or a new reality, because our world is changing and we need to be on time with it. So natural means that it's not synthetical. Natural, it seems that we have some control. It's related more now with the sustainability, circular economy, upcycling, ethical, recycling, green, whatever, clean beauty, zero waste. Even the COVID-19 included us or make us think about more in the climate change when we were all in lockdown. So we are, it's not a trend, is something that we must do to try to remediate what we have done to this world. So it should begin with a trend, but now is more than this. The new concept related to clean beauty, and when I'm talking about this type of concepts, is always related with the final product that the consumer will use, not only with the ingredients. So clean beauty started also to simple formulations with clean, with the safe natural ingredients, but is more and more gaining more strength as a sustainability, transparency from the companies to the customers, to have more trust for the customers. Customers now want to know not only about the product, they want to know what is the history from the product, where the raw materials came from, what was all the process to make until we arrive to the final product for them to consume. And later on, we will see that it's not till this step. We will consumers want to know what happens to the product after consumed. So clean beauty is starting to again like a trend, but is gaining more and more strength in the market. Another thing that is driving the consumers is not only the ingredients, it's all in all the process. If all the companies, since the raw materials, manufacturing, that are involved in producing one product, if they have a social responsibility, if they are focused also on economic progress, maintain the natural balance of the planet. So this is all linked. Besides raw materials and the process, we have also the packaging, the 5R principle. The packaging is I won't say a issue, but it's in the embryonic stage, and maybe in the formulation stage we can see more advances at this moment, but we are moving forward. There is still there are appearing more solutions now. So what consumer wants, what consumer expect from one product? Just when a consumer buys a product, is not only to see, okay, I have a body hydrating cream or whatever. They want to know all the story and what will happen to the environment and to their health after using this product. So besides all the claims, all the demands and the expectations that customers have about a cosmetic product, as a product developers, we need to pay attention to these questions and develop according to these requisites. So they want to keep the efficacy, they want to be safe, they want to product has the same or higher quality, being sure with the certifications, tests with the animals to avoid. So all expressed in the label. So with so many checklists, my checklist so big, what to do? So what are our challenges as a product developers and at this moment is starting all with an idea, and there are a lot of challenge through that came from that new idea, new concept of a product or to solve an issue, and it seems a long way to arrive to a final product that fulfills the expectations of the consumers. So the main, for me, from my point of view, the main challenges are regulation certifications into the industry and what is the message that is passed to the consumers. The ingredients functionality, if we need to change to a new alternatives, maybe we are reducing some functionalities that were used to with the all previous non-certificated raw materials. The production process is also something to pay attention, and of course the packaging and in the way that the product is dispensed. So we are going through these points. I'm not going to speak this in an exhaustive way, just to give some points of what is now and what we are dealing now. Natural or natural derived ingredients, it depends if they are really came from the nature or they suffer some chemical modification. And how we see these is like, it depends if we are considering a Cosmos certification, if we are follow the ISO, if we are following the NATRU. I realized that recently we have an upcycled certification standard version 1. So this is all the information that came from, to us, that is expected to be present or to be transmitted to the final consumer. So when final consumer wants a Cosmos certified product, we need to go to the raw materials, be sure that all the raw materials are Cosmos certified. So there's a lot of limitations and challenge. Going back a little bit more about the natural concept. As we saw previously, we have different types of certifications, but we have also different definitions of natural. There's no official or global definition of natural. And this is a difficulty that we have in the product development. If we are developing a Cosmos, perhaps the criteria used in the ISO is not enough to proceed with the Cosmos formulation. This is an example. For instance, ingredients functionality. So this type of certification of the ingredients that will also certificate the final product has some criteria. And not all the raw materials that we have available could be certified because they are restricted according to the manufacturing process, origin, whatever. What I mean is that the source of the raw materials is really important, but not all sources could be used to be using this type of certification. So what happens is that we are not we haven't all the raw materials available to achieve the same performance that the final consumer knows for the regular cosmetic products. Although there was being increasing in this type of ingredients, the offer that we have. Common natural ingredients that we normally use a long time ago is the essential oils, plant-based extracts, vegetable oils, some of them to give some fragrance or alternative to synthetic fragrance. So although this is quite new or quite recent because it's has some years, this type of ingredients we already know, or at least we know how to work with them. But sometimes what happens is that the performance is not what is expected, because we can face some issues, or for instance, if I'm used to work with a type of surfactant, if I go to a natural Cosmos certified, maybe I would have the same performance, or the colorants or the dyes, maybe the synthetic are more controlled because during its production, but maybe in some options we cannot control the heavy metals. The preservatives also referred in the previous presentations by Dr. Ana Oliveira. The preservative free is not it's like a wrong idea. We should always to use preservatives according to our regulation. Fragrance also could have some issues. Some raw materials could have some problems with the perfumes, with the odor. With the color, also previously referred. So the part of the aesthetic and performance is not of being natural, be more easy to work with. Another thing I would like to speak about is the RSPO, the Roundtable Sustainable Palm Oil. That's common ingredients like glycerin or the main of the surfactants are sourced. Here in the company, we have this attention in order to use the sustainable palm oil source. We have also in the market similar sustainable roundtables for soy, cocoa, because the meaning of being natural, it doesn't mean that it's sustainable. It couldn't, because if you want to have huge quantities, or we need to have huge quantities, not only for the cosmetic, but mainly in this case, SPO for the food industry, the demand is so high that is not sustainable to deforest to continuous produce the palm oil. Then another point that I would like to say is the production process. Production process is also something that is forgotten, but is important to say about the part of sustainability. Is not only about the raw materials and the sources, is how we are using, how we are mixing them, are we consuming too much energy, are we spend too much water. Water is precious in these days. So this type of thinking is in our mind also when we are developing new products. Relating to the packaging, our focus is mainly here in Valcambra, aerosol. I believe in some years ago, decades ago, aerosol were seen like or demonized like the best or the worst thing to the planet. Aerosol suffered a lot of evolution. The technology evolves really fast. So they have a lot of advantage. You can apply a product with a contactless or self-dispensing. You can apply the product would not even touch the inside of the product. The product is protected inside of the can. You can have different sprays performance. You can have different types of foams. And of course, we have the inhalers, medical for the asthma. Aerosols are now being the propellant is being substituted the LPG for the nitrogen, for instance, or another compressed gas. So the knowledge that we have about these technologies are the key to develop new products and to propose new sustainable options to the customers. So after saying all of these, returning now a little bit more at Colab, we are in our development focus in the 4 main areas. We have the sustainable packaging dispensing systems. We have natural propellants and ingredients. We are focused also in dry waterless products, and also the new reality post 2020 or post pandemic. I have here some of our innovations. We have the first Cosmetic product in the market. We were the first technology to space in the aerosol. We produced the Turbo Tango, and we have the oil foam, or the natural foam with the carbon dioxide. So this is some of our achievements that we made with this type of products. I have also more to show to you. So of our innovations, we have the compressed technology. Imagine if we have a big can, but we can reduce the size because we can dispense the same amount of product, but with less propellant. This will enable us to reduce the size of the can, and this is, in terms of packaging, more sustainable. With the same duration. For instance, you have a small can that will have the same utilizations that would have one big can. So this is one part of our innovation. We have also, like I said before, switching the LPG. LPG is the compressed gas, the liquefied petroleum gas, that is substituted for the compressed gas. And this will allow us to reduce the VOC, volatile organic compounds, and to offer products more safe. But just for curiosity, these type of products could also be Cosmo certified. That's one of the certifications that is more tricky, difficult to get into offer to the customers. So we have this type of products to offer. This is just a small overview of these of our work as a product developers, my personal view about the natural trend in the product development. Thank you for having me here, and I'm available for any questions that we have. Thank you.
Okay, thank you very much, Isabel, for your excellent presentation. Let's check out the questions that our participants have to ask you. Okay, I think we don't have any questions, but I have one curiosity. When we talk about natural products, we talk about using nature to extract the ingredients, the natural and functional ingredients, to enrich the final products. So my question is, do you consider this point sustainable?
It depends on the source. It's like the example of the RSPO, the palm oil. The idea is not to exhaust all the sources of one raw material until it ends. So you are using natural, but it's not sustainable. So the idea is to close the circle. Like we heard today, the upcycling, or if we go to the nature, take this something as a raw material, we can give back in another way. So in this way, we don't take anything from nature. This is the principles of the clean beauty and sustainability is more than the concept of the natural, is more than that.
Okay, thank you again, Isabel. And now we advance to the closing section with the coordinator of the Centro de Investigação de Montanha, Dr. José Alberto Pereira, and Chair Lilian Barros.
Thank you, Christina. Isabel, I would ask you now, please, could you interrupt your presentation? Thank you. So I would like to correct my colleague, Christina Kaleja. For the closing session, we will have our IPB's President for Research and Innovation, Dr. Tiago Barbosa, which I ask if you could please turn on your camera at this point. Thank you, Tiago. And I would also like to thank you very much for being here with us today. For us, it's a great pleasure to have you in this Congress and to have research and innovation, which we are dealing with during this conference since yesterday, trying to show the outputs of science that is being held during this problematic situation and pandemic situation, which difficults a lot the work and scientists like we are to have their work overcoming all these problems. So I thank you I thank you for being with us here today, and I will now let you say your words and share the screen so you can comment on this. Thank you.
Yeah, thank you so much. Hopefully everyone is able to listen me clearly. First of all, it's a pleasure to be here. I believe we are a little bit behind schedule, so I will try to shorten down a little bit my closing remarks. Okay, so yeah, this is quite unique, isn't it? So first, I should say good morning, good afternoon, or good evening, depending on where are you from or in which time zone you are attending this closing ceremony and this full event. And as I was saying, this is quite unique. This is very unique because just a few months ago, it would be unthinkable, this kind of opening remark, isn't it? Greeting attendees spread over so many countries. So firstly, I would like to congratulate the organizing committee for taking this challenge, as Lilian was saying, and it's a huge challenge putting up this scientific meeting in such unprecedented times. So my warmest congratulations to you, Dr. Lilian, as chair of this event. And please, Lilian, forward this note to all members of the organizing and scientific committees. I believe they put a lot of hard work so that you had a very nice and outstanding event. So not surprisingly, the Amount of Research Center positions itself as a research driver here at IPB. This is a very, very important research center for us here. And this is for quite a while. And not to mention, obviously, the important role that has been played more recently by MORE, the Mountains of Research Collaborative Lab, as well as Aquavalor. And my understanding is also that several European funding agencies, several stakeholders are keen to promote and facilitate research on topics related to this scientific field, to natural products, its application on health, on cosmetics, and food. So yeah, I believe you have very exciting times ahead, even though we are struggling right now. We are coping with this pandemic that puts a lot of pressure on everyone. My second note, it's a note, and I would like to note the good traction that this Congress had. I'm told it has over 400 participants and more than 220 talks. So I'm sure these were days of intensive learning, networking, and sharing among all of you in this virtual forum, isn't it? And well, we have to understand that the academia landscape is reshaping rather quickly. And just to showcase my standpoint on this, soon, very soon, hopefully, there is an opportunity to hold hybrid scientific meetings. And participants like yourself, just like myself, can choose, we'll have a box to tick. I want to attend this conference on site or online. Well, that said, we are all on the same page if I argue that one misses so many things to attend a conference on site, isn't it? And well, so hopefully in the coming future, you are going to be again together, but this time face to face. And from our side here in Bragança at IPB, we are looking forward to host you here in Bragança as soon as the pandemic goes away, as soon as we resume the normal engagements, or at least an updated version of what's going to be the new normal. Because as I was saying a few minutes ago, the pandemic is putting some pressure on us when we talk about science and when we talk about innovation. But with the pandemic being under control, hopefully we can again resume full steam and work as we should be doing it. And so this is my last note. Thank you so much. I hope to see you soon here in Bragança. And I pass my time back to you, Lillian.
Thank you, Tiago, for your kind words. They are very motivating. I hope that, yes, soonly we can have these type of conference on site, because it's really easy to have it online, of course, but the contact with the human being in contact with the people is always very different than online. So hopefully this pandemic situation will pass. And in our second edition, we hope it could be on site and have a double, like you were saying, a double section on site and online also for this for a possible second edition of this conference. So thank you very much for your kind words, and I will now continue just with some brief words to all the participants. And because we are now concluding this first edition of the Natural Product Application Online Congress, I just want to say that this conference was organized by the Mountain Research Center of the Polytech Institute of Bragança. It was made in collaboration with Colab More and the Seat Aquavalor, which I also thank for their collaboration in organizing this conference. For us, it was a great pleasure and an honor to spend these 2 days with all of you. And in my opinion, it was a very successful conference, even though being online. Therefore, on behalf of CIMU, the center, the Mountain Research Center, the NPA team, and all the collaborators, we would like to extend our most sincere gratitude to all the participants, experts, and assistants in joining efforts to be with us during this journey in science, of course. Today, we are all proud because we accomplished the objective of this conference, which was to join the industry and scientific community related to natural products, especially those in the fields of health, food, and cosmetic, and disseminate the latest updates and also the current works that we are performing nowadays. We are very proud of the numbers we obtained for this conference. because thanks to all of you, we have registered 481 assistants, had 9 keynote lectures, and 1 special invited communication. We received 211 abstract submissions, from which the scientific community had a hard time selecting 18 oral presentations and 18 pitch presentations. And there was also 166 posters that were displayed online in our webpage, and which all of you could consult now, and it will be online for a couple of days also. We will also have a book of abstracts with all the information gathered in the conference and with all your abstracts. This will be sent by the end of next week, I hope, of course, and will also be available on our website. Due to the digital format of the conference, we were able to gather some numbers for your information. For example, from our social media diffusion, we reached 2.2 thousand people on Facebook and Instagram. And on the YouTube streaming sessions, we gathered another 1.8 thousand viewers until today, okay? Because they will be online and everybody can consult and see all the sessions made during this time. So during the 3 sections, we had a median number of around 147 assistants on the YouTube streaming, which is really, really good. So therefore, due to this success, and as I already mentioned, we will try to have a second edition in 2002, probably also in February in this month, to have a second edition of this Natural Products Application Conference. We hope it could be in the double situation on site and online. And like this, we can have a nice conference like we did today. And of course, if it's in its presential form, you can also visit our beautiful city of Bragança. So we ask you to please keep tuned to our social media, Facebook and Instagram, to have more information regarding the second edition. I want to take this time also to thank the organizing committee in the names of Dr. Alexandre Gonçalves, Dr. Maria José Alves, Dr. Rui Abreu, Dr. Ricardo Calhella, Dr. Soraya Falcão, Dr. Bruno Melgar, Dr. Bruna Ribeiro, Dr. Carlos Shirashi, for their collaboration in organizing this conference. Though I have to take this time also to give a special thanks to Bruno Melgar, who has followed all the stages of the conference and helped me a lot during all this process as the chair of this conference. I would like to give him a note. I think he has never answered so many emails in his life. He tried to respond to all of you in the due time. Of course, that since we received so many emails, sometimes it was difficult. To the scientific committee, Dr. Rosana Peralta, Dr. Manuela Pintado, Dr. Jesus Simal, Dr. Paola Perugini, Dr. Eliana Soto, and Dr. Joana Amaral for their collaboration in selecting the most interesting works that were presented during these 2 days. Also, to the monitors, Dr. Soraya Falcão, Dr. Maria Inês Dias, Dr. Felipa Reis, Dr. Sandrina Heleno, Dr. Márcio Carocho, Dr. Carla and Dr. Pereira, and Dr. Cristina Caleja, for their help and for following all the works and adding such interesting questions to all the presentations made during this conference. To the Innovation and Web Project Center, especially to Evandro Alves and to Dr. Luís Pires for their help on the online streaming. To the keynote speakers and to the invited oral communications for accepting this challenge to disseminate science in this time of crisis that we are going through. And finally, but not least, to our special opening guest speaker, Professor Isabel Ferreira, Secretary of State of Inline Improvement, as also to the institutional delegates, Professor Orlando Rodrigues, President of IPB, and to Professor Tiago Barbosa, our Pro-President of Research and Innovation. So I hope I didn't forget anybody, and if I do, I ask now for my forgiveness, because I think I did not miss anybody that helped during the elaboration of this conference. So to continue, and just very briefly, 2002 was a great challenge to all of us involved in science, of course, but not only. And 2021 is also showing some great challenges. Although, in the words of Carl Sagan, which I now quote, "Science is not perfect. It can be used. It is only a tool, but is by far the best tool we have. Self-correcting, ongoing, and applicable to everything." So, from these words, we need to keep improving this tool in our specific areas in order to overcome all the current challenges we are facing. We hope for all the assistance to keep growing the connections created in this conference, and we hope to see you soon in our second edition of the Natural Products Application. So thank you all for your participation. Stay safe and see you soon. Bye.